Our next speaker, I'm very pleased to introduce, is Professor Daniel Olive. Daniel is a professor of immunology at Marseille University in France, and he's the head of the Immunity and Cancer Research Team at the Institut Pauli Calmette Lab and director of the immunomonitoring platform at the first IBISA, dedicated to cancer immune monitoring. He's been a pioneer and leader in the co-signaling field since 1990, and his work is dedicated to tumor immunology with a major emphasis on innate immunity and co-signaling molecules. He's been really pivotal in a number of major breakthroughs, including the identification of signaling pathways involved in CD28 and ICOS co-stimulatory molecules, identification of molecular mechanisms associated with NK cell impairment in AML patients, and first-in-man clinical trial using anti-KIR monoclonal antibodies in AML patients, and deciphering of ICOS ligand interaction in the regulation of Tregs and lymphomas. So Daniel, thank you very much indeed for being able to speak to us today, and we're very much looking forward to hearing your presentation on characterization of Vδ2 gamma delta T cell populations in breast cancers. So thank you very much.
Yeah, thanks for a nice introduction. You hear me? Is that okay? Okay. So thanks for a nice invitation, Jeremy. I appreciated it. So I will try to present data that are different from one that are on the previous list that you listed, and that are more dedicated to a population of cells that are not found in rodents but are found in humans, and that are associated with the prognosis in patients. I will focus today on breast cancer and just to introduce the field. So we are moving from COVID-19, previous presentation, moving to cancer. So there are some issues that are known in general regarding breast cancer. The first one is that metastatic breast cancer, although rare, fortunately, are with very adverse prognosis and poor survival. Among them, the ones that are called triple negative breast cancers are difficult to treat using the classical chemo plus surgery. And so most of the patients die at the end. And the other way around, due to the classification, there are over-treatment in some patients. So it's important to get some more insight in the classification of this patient to identify the parameters important. So immune parameters were under the limelight recently on breast cancer for different reasons. The first one, people identifying a subgroup of triple negative breast cancers, high lymphocyte infiltration. That was surprising initially. And we know that the lymphocyte predominant breast cancer have a more favorable prognosis. And this infiltrate in situ is associated to prognosis and predictive. And this provided the way to move to clinical intervention using a classical anti-PD-L1 antibody and anti-PD-1. And what was shown recently, last year, is that you could improve the prognosis of a patient with this very aggressive cancer with a combination of chemo plus the anti-PD-1 antibody. So it means that there is room at the present time to evolve these immune parameters. Here I was talking mainly on CD8. So this slide taken from a review for Van Keer and all is very nice and simple because it outlines what are the different types of immune cells we are facing in cancer and infectious disease as well, of course. Is that on one hand you have the classical innate cells, including NKs and ILC, for instance, and the classical adaptive, meaning B cells, T cells. But in between, you have cells that are innate-like. The reason why they're called like, because among others, they have expression of BCR, for instance, or B1 as a B cells, or a TCR that is different from the one from the alpha beta cells, but they are a TCR. And among these ones are the gamma delta. The major difference is that they are both the capacity to be activated and act rapidly, like innate cells, but also have a recognition of some determinants that are different from the peptides presented to MHC, and I will present it just after. So gamma delta are under the limelight for a few years, and it came back in 2015. Initially, it was shown that they were important in mice, in protective, in cancer models. Then there was a seminal publication in the 90s showing that after bone marrow transplantation, they were associated with better prognosis. And what was really astonishing was this paper in 2015 showing that if you do pan-cancer transcriptome analysis, meaning most of the human tumors, solid or leukemias, the markers, the human markers that are coming first were gamma delta, then others that were more expected, TFH and CD8, but they were coming first. This was a surprise. Or is it the end of the story? No, because the story is a little bit more complicated if you put it from the 80s when the gamma delta were identified till 2020. Let's say that there are different things that came out. Early 90s, people identified gamma delta in breast cancer with TILs. These cells were able to have some in vitro function against breast cancer cell lines. And the ligands of the Vδ2, I will come to this on the next slide as well, were identified, are present in breast tumors. And next step was to show that these cells, in general, in patients with breast cancer, was poorly active. So people try to expand the cells and to get some immune intervention with cell therapy using hyaluronic acid activated gamma delta cells, and to show that there were some insight and some interesting features in terms of responses in some patients, but it was not overwhelming. Otherwise, it would be the therapy used now, but there were some signs. Where things were more complicated, and is where I'm turning progressively, is that what people identify is that, like T cells, there are regulatory gamma delta cells that were found in breast cancer. And it was shown that at the same time, gamma delta cells are useful for predicting the poor outcome, it seems to be related to this factor. And so we were surprised by 2015 data, of course, based on what was shown previously. And among the different things that came out is that in addition to the regulatory gamma delta, there are also IL-17 producing gamma delta that are associated to poor prognosis in mice and also seem to be in the same way in humans. Studies continued, and last 2 years ago now, what was shown is that we could reconcile in part the story if you investigated what the feature of the gamma delta, and showing that the one producing Th1 cytokines were associated to good prognosis. Even more recently, a marker of gamma delta regulatory was defined as CD73. So meaning that we are more in line with what is shown in human for T cells, where we have different subsets associated to a prognosis. So one thing, so I'll continue with what are the receptors and ligands that are involved in the function of the gamma delta. I would talk mainly of Vδ 2, and the way the TCR work is by recognition of different types of compounds that make a connection between infectious disease that we discussed just before and cancer. meaning after infection by intracellular bacteria like TB, you have production by a cycle called a Romer cycle of compounds that are phosphorylated compounds that are able, the MAPP, that are able to activate the gamma delta. If you are going now to human cells, you have the mevalonate pathway leading to cholesterol at the end, and within the mevalonate pathway, there is a generation what is called isopentenyl pyrophosphate or phosphoantigens that accumulate in tumors. There is a dysregulation, accumulation of this compound. So in these two settings, infectious disease and cancer on the other side. So the story of recognition is a little bit complicated with the gamma delta. So they have a TCR, I told you. They recognize phosphoantigen, indicated here as P, and there are different receptors that are important to elicit the function. And the ones that are critical are called the butyrophilins 3A and 2A. And we identified in 2012 between 3A as the key receptor. Otherwise, they have a function of innate effectors, production of cytokines, and ability to kill once they recognize these phosphorylated antigens. Butyrophilins were identified a long time ago, more or less 30 years ago, by Peter Linsley. These molecules resemble the B7 family members that are important for plenty of different functions. And what Peter identified is that this molecule was rather weird because it has a binding module, B7-like, with the two Ag superfamily members, but the intracellular domain was composed of what is called B30.2. And B30.2 is a molecule that is known for a while, more or less 100 molecules possess B30.2, important for inflammation and innate functions. Innate functions, for instance, for decapsulation of HIV, or there are inflammatory diseases where there are mutations of this domain. So it was a mystery. And if you come now to the phylogeny of the molecules, so there's a large family with 14 butyrophilins found in mammals, with very major differences between rodents and primates, because only 8 out of 14 that are conserved between both. And among this molecule, I'm coming back now to BTN2A1 that is found only in humans, not in rodents, and BTN3A1 that is found only in humans, not in rodents. So these two molecules are critical, have evolved and are found in primate, of course, are the same to elicit and control the function of gamma delta. So coming back now, after this introduction, what are the data? I will be rapid on the data. So the key issues, you have understood that the prognosis of gamma delta is controversial in different tumor settings with function for delta 1 that are either Th1 or regulatory. But in general, the delta 2 are mainly pro-inflammatory and cytotoxic. So the information we don't have in cancer, human breast cancer, is what is the status of these cells and the interaction with clinical parameters. So the study was done at a single cell level. It's a cytoph study, so it's a mice cytometry study. The goal was to identify the cells and to associate them to the markers, clinical markers. So there was a large list of markers that were investigated. I will not go into detail for sake of time. And just to indicate that we took patients not treated, so at entry at the hospital, at early stages. And the goal was to do what we call a liquid biopsy. That is really to investigate what was in the peripheral blood of the patients. and to compare it to what was in the tumor. A few molecules that are known to be for a function on gamma delta, for instance, PD-1, that is a well-known molecule, of course, regulates the function on gamma delta, that is overexpression prevent some of the production of cytokines. CD57, that is normal on T cells, also expressed on some gamma delta subpopulation, and more of the one that are associated with what we call replicative senescence, and with diminished function in terms of cytotoxicity. IL-7 is key, like T cells, to permit to have naive cells and that develop and proliferate. And 56, that is a marker of T cells and mainly NKs, is more present on activated gamma delta and permit to increase the function. They are less function and less understanding what the role of LAG3, TIGIT, and TIM3 on the gamma delta. So I skip this one and go to the data. So what we have done, more of the data are put together. These are bioinformatics analyses. I will show some data afterwards. So this is the integration by what we call between group analysis, where you investigate all the population that the CyTOF can provide on all the immune population. So with different panels, we investigate all the markers on gamma delta as controls, CD4, CD8, NK, of course, just to have a view. So what is clear from a between group analysis, if you compare the healthy volunteers to breast cancer, you see that there is a different stratification. To take an example of top 10 variables, for instance, in healthy volunteers, you have more naive gamma delta. In contrast, in breast cancer, you have more activated and differentiated gamma delta. If you further investigate this, still by this between group analysis, to investigate what is more associated and enriched in breast cancer present to healthy volunteers, you have here a more simple way to present it. So among the different markers that came out, the ones that are more associated to breast cancer here, in addition to the one that are expected, like CD8, of course, that is expected, NKs are expected as well, but gamma delta, delta 2 were highly associated and enriched in breast cancer. To continue this between group analysis from the site of data, we now stratified N+, meaning patients with the involvement of lymph nodes or not, N-. And the one where you have the more enrichment in terms of vitals, the one that came out are the gamma delta, delta 2, and a subgroup, what we call terminally differentiated, and the one that are senescent, that came first in the study. Is shown here in more details, and still in terms of percentages, in terms of the markers that are more associated to the involvement of lymph nodes, correspond to 37% the gamma delta of TAMRA and CD57. Skip this one. So I show some data, so it's helpful to put all together the data. So once you have your data obtained by this type of analysis by CyTOF, you then doing what we call a t-SNE, that is to get only 2 dimensions to investigate what are the different subgroups that differs between here, breast cancer with no involvement of lymph nodes, of involvement of lymph node. And what you see, and then I will show the markers after this, is that there are different groups that are enriched. Specifically, I will come mainly to these ones called 28, 26, 27. And among the parameters and molecules that were more expressed by these cells, you can detect CD57, CD16, as well as CD56. And if you go in more details in the different cluster and investigate their weight in terms of prognosis and percentage, what we can see is that the ones that accumulate more in breast cancer with lymph node involvement, again, everything is liquid biopsy, nothing in the tumor at this stage, correspond to these cells that are thermally differentiated. Now we have numbers that you know, of course. So RA positive, and this type TEMRA classical, plus or minus PD1, for instance. So meaning that this 57 on the other one, that these cells tend to accumulate and be associated with the prognosis. So to sum up at this stage, what are the data from this part at biopsy? So we can detect these alterations in addition to what is known from CD8. So there are additional population and that are highly associated prognosis. So the Vδ2 are associated. Their phenotype is more exhausted senescent cells. And what is ongoing now that we're doing is a paired control, paired site of analysis of patients' samples, blood and tumors. And just to provide an example, because I'm running out of time in 1 minute, Jeremy, you interrupt me whenever you want, of course. So just to give an example on the first patients we investigate in the tumor now from the paired samples, and what is in green, again, these are t-SNE, so 2D representation of the data. In green are the normal breast tissue, and in red, what corresponds to a cancer. And so you notice the stratification is totally different from CD8, that is known, but also for Vδ2. And among the differences that are shown here on this very simple TMF representation, you have another representation in the tumor of this type of cells, meaning the one that express CD69, 28, IL-7, so for naive, as well as CD27 and CCR7, whereas group 5 and 6 tend to have CD16, CD56, and CD45 array, so more TEMRA-like. And now, if you investigate these different groups, cluster 1 and cluster 6, what we can see is that we tend, in contrast to what we have in the peripheral blood, to have more early effector memory or central memory cells in the tumor, whereas the TEMRA tend to be decreased, meaning like they cannot enter or they are blockade of their differentiation in the tumor. So it basically is what we're investigating right now in the setting. So it's a more detailed data. And just to present the two people who did the work. So Stéphane Fattori, who is a PhD student, and Anne-Sophie Chrétien, who is associate professor in the team. So I will be happy to answer to your question. I have additional backup slide if needed, and I can go in more details. So Jeremy, I'll let you organize.
Wonderful, thank you. Thank you very much, Daniel. I really appreciate it. It's great to hear that amazing piece of work. So I'd like to open the floor for questions. Does anybody have any questions for Professor Olive? While people are waiting, you can raise your hand or raise your hand or put it in the chat box. Daniel, just could you, from a point of view of actually sort of moving this into a sort of a diagnostic, is this something, what sort of stage has that got to in respects to sort of translation of this identification? Is that something which is going to be sort of simple enough to be able to translate into a diagnostic tool?
I think so. There is an example. I was hesitating to present it as well, because we are working for more or less 20 years on NKs, and among the NKs there are markers associated to a prognosis, and some of the markers, for instance, are used now in my hospital that are associated to the response, and it is important for in leukemia to predict whether we are moving or not to aggressive treatment or bone marrow and allo stem cell transplantation. So this is already done in this setting. The idea is due to the fact that it's complicated to get samples from tumors. Of course, we are doing this when people enter the hospital, but doing iterative biopsies is complicated. And the goal is the reason when people are doing ctDNA, for instance, for the tumors to predict it, and mutations within ctDNA, is to have some of the markers that are also associated to prognosis. So my guess, but we need to have what we call validation cohort. So this was exploratory at this stage on a low number of patients. Now we are increasing the numbers in a clinical trials that is ongoing in our hospital to have more patients to get a validation cohort.
Great. No, thank you for that explanation. That's great. Any other questions at all? I don't think so. I can't see any. Well, thank you very much indeed. Thank you, Daniel.
Thanks, Jeremy.